Hello everybody, in this video lecture we will cover mitosis. So this is a PowerPoint that I will use. Okay, let's go ahead and begin. Mitosis plays important roles in the lives of organisms. Mitosis replaces damaged or lost cells, permits growth, and allows for asexual reproduction. So those are three functions of mitosis. So here you can see how mitosis are involved in a cell replacement, replace damaged cells. Right, so here's what's shown to you is a human kidney cell going through process of division, right? So some cells are damaged, we wanna replace them with new one. Here, growth via cell division, that's a human embryo. All human and other animals begins with one single cell, with a zygote, then the zygote divides, forming the whole bunch of new cells, right? So that's the growth and division, cell replacement. And here's on this picture, you can see asexual reproduction. In asexual reproduction, single-celled organisms reproduce by simple cell division. There is no fertilization of an egg by a sperm. Unicellular organisms like this amoeba shown here, they divide by mitosis, so they just split into two parts, and each one has identical, well, nothing is identical, never identical, but very, very similar DNA, because mutation still happens. But here we have clone, two daughter cells that are clone of a parent cell. Some multicellular organisms, such as sea star, for example, can grow new individual from fragmented pieces, right? So that's also a sexual reproduction. So again, this new organism has the same DNA as a parent organism. Growth of a new plant, for example, over here from clipping, is another example of asexual reproduction. So cell division happens during mitotic phase. Mitotic phase is a part of cell cycle, and it includes mitosis and cytokinesis. So the mitotic phase is a multi-step process during which the duplicated chromosomes are moved to opposite poles of the cell, and then the cell is divided into two new identical daughter cells. So over here on this diagram, you can see mitosis and cytokinesis. So interphase, during interphase, specifically this is G2 phase of interphase. We already see that the centromeres are replicated, but inside the nucleus, you don't see chromosomes, you see chromatin. And then cell enters prophase, metaphase, anaphase, telophase, cytokinesis. And there are many intermediate stages like proto-metaphase. It can be like early anaphase, late anaphase, early prophase, because that's a continuous process. That's why there are so many intermediate phases, but the major are prophase, metaphase, anaphase, telophase. That's a nuclear division, and then cytokinesis, division of cytoplasm. And you can see how cell, now you can see chromosomes inside the nucleus. Now those chromosomes line up in an equator or in the middle of a cell. Those yellow lines are mitotic spindle. So those are special proteins that attaches to chromosome, and then they pull sister chromatids apart. Those sister chromatids, when they separated from each other, now they chromosomes, individual chromosomes, so they migrate towards the poles, and two new nuclei are formed, and then two new cells are formed. So the first portion of the mitotic phase, mitosis, is composed of 4 stages which accomplish nuclear division. The second portion of mitotic phase, called cytokinesis, it is physical separation of the cytoplasmic components into two daughter cells. So this pie diagram represents cell cycle. Everything shown in gray color is interphase. cell is not dividing during interphase, but cell replicates its DNA. And then mitotic phase include mitosis and cytokinesis. Now, mitosis has these four major phases: prophase, metaphase, anaphase, and telophase. So here's G2 of interphase, and then we enter prophase, metaphase, anaphase, telophase, and cytokinesis. Mitosis results in a division of the cell nucleus. Interphase, that part, this is G2 phase, right? Cell is not dividing yet, but in G2, all chromosomes are replicated already. So here's we can see chromatin still inside the nucleus, not chromosomes, nuclear envelope attached, and centrosomes. Centrosomes are, this organelle over here with centrioles. So inside this yellow parts are centrioles, and the whole organelle is centrosome. Now, what happened during prophase? In the prophase, mitotic spindle formed. So this is the early mitotic spindle, and those are late mitotic spindle, right? So those are special proteins that grow from the centrosomes, and centrosomes start moving towards the poles. So mitotic spindle formed, centrioles migrate to cell poles. So here you can see they already migrated. Chromatin condenses into visible chromosomes. Nuclear membrane dissolves. So over here, we still have nuclear membrane, but in this prophase, kind of like late prophase, now you see only fragments of nuclear envelope, and metabolic activity decreases. After prophase, cell enter metaphase. In metaphase, duplicated chromosomes form a single line at the equator between centriole poles. So here's chromosome in the middle, right between these two centrioles. Now this mitotic spindle is attached to the centromere. It's called kinetochore over here. So it's attached to the middle of each chromosome. In anaphase, duplicated chromosomes separate, and daughter chromosomes are pulled towards poles by microtubules. So those are microtubules, part of mitotic spindle, and they contract and pulling these chromosomes towards the poles. In anaphase, chromosomes look like letter V. That's anaphase. And here's the telophase, so metaphase, anaphase, telophase, and cytokinesis. Cytokinesis begins at late anaphase, and then it's continues with telophase until cells completely divided. But in telophase, we have processes that are reverse of prophase. So mitotic spindle disintegrate, nuclear membrane reforms, and chromosome uncoil and revert to chromatin. Right, so you can see reformation of nuclear envelope, chromosome again became loose, returned back to chromatin, and no mitotic spindle. Cytokinesis usually begins during telophase. It says telophase over here. Okay, so in some books you will read it even starts at the late anaphase, but we can live with telophase. And cytokinesis divides the cytoplasm and different in plants and animal cells. So on this picture, you can see animal cell cytokinesis and plant cell cytokinesis. But result is the same, two daughter cells. Those daughter cells are identical to each other and to a parent cell. Now, let's look at cytokinesis in animal cells. A ring of contractile protein immediately inside the plasma membrane at the equator pulls the plasma membrane inward. So if you look over here, that's a metaphase. This is anaphase. Okay, so on this picture, you see that's correct because cytokinesis begins at anaphase. So you see those proteins, those are contractile proteins, and they start pulling this membrane at the equator inward. The inward pull of the plasma membrane produces characteristic cleavage furrow. Now you see this like indentation and this indentation, this is called cleavage furrow. When the cleavage furrow reaches the center, it's pinched apart to form two daughter cells, right? So we have a contractile protein, they are inside a cell, but they at the equator, and they start pulling this membrane towards the center, right? And making this cleavage furrow deeper and deeper and deeper. And at the end, two daughter cells are pinched apart. In plants, during telophase, membrane-enclosed vesicles derived from Golgi apparatus and migrate to the center of the cell. So there is no cleavage furrow, there is no contractile proteins. But what happened inside the cell, you have Golgi apparatus, right? So here will be a vesicle from Golgi apparatus, and they will migrate in the center of the cell. Vesicles fuse to form tubular structure, which form two layers of plasma membrane. That's called cell plate. So the new plasma membrane is formed from these vesicles right in the middle of a plant cell. The cell plate continues to develop until it connects the existing cell's plasma membrane. So here's vesicle from Golgi in the middle, forming this cell plate, and it's continued to develop until it touches this yellow part at existing cell membrane. Then both daughter cells secrete cellulose to form their new adjoining cell wall. Now each of the cells starts secreting cellulose, right, on the outer surface of the cell plate, forming this cell wall. So yellow is cell membrane and green is cell wall. So we have cell plate during cytokinesis in plants, and we have cleavage furrow in animal cells. Okay, so this was the last slide for this lecture. Thank you for watching, and I hope it was helpful.